everyone. My name is Sushesh, and the title of my talk for today is Development and Validation of Microdialysis-Based Discriminatory and Biorelevant In Vitro Release Testing Instrument for Complex Ophthalmic Products. Now, let's take a look at the challenges and the complexities along with the scope of research that ophthalmic drug delivery provides. So, as we all know, eye is a complex organ. The anatomy of the eye is complex. It provides a very limited residence time for any formulation that we put in there. It has a bunch of ophthalmic barriers, and also the tear film dynamics in the eye are very variable. So coming to the formulation challenges, the sterility, stability is a challenge, dosage accuracy, container closure system, manufacturing processes, and these are a bunch of challenges that ophthalmic formulations have. So now, demonstrating bioequivalence for any ophthalmic product it's actually very difficult and pretty invasive to perform a PK endpoint study. So here comes the scope of our research is to develop and validate an in vitro release testing instrument and a method which is robust enough and sensitive enough to not only prove the sameness but also catch hold of the pitfalls of any differences in the formulations. Of course, this comes along with a bunch of advantages, which is cost cutting, reduce the economic burden on the healthcare system, which would also accelerate the generic product approval. For a generic product development, especially for that of ophthalmic products, besides the IVRT technique, there are a bunch of other critical quality attributes, which when measured in an in vitro setting, if they are same for that of the generic product, it is expected that both the generic product would actually perform same or similarly in an in vivo setting and also in a clinical setting. So there is actually this is in par with the guidance that is given by the US FDA, which talks about the in vitro option for generic product development of an ophthalmic drug product. Now, actually, how we approach the problem to develop and validate our method is by formulating a bunch of different formulations. As you can see here, RLD is a 0.05% strength difluoretinate emulsion product. The F1 formulation, it's a Q2 equivalent formulation to that of the RLD, which means it's both qualitatively and quantitatively equal to that of the RLD. F2 is a half strength product. F3 is a double strength product. F4 is also a Q2 equivalent formulation, but there is a formulation technique variable there. And F5, it's actually not an emulsion, but it's a micellar product. You will actually understand why we chose to formulate all these different types of formulations in the upcoming slides. So here are some of the critical quality attributes that we try to measure, such as particle size, PDI, viscosity, and pH. If you can see here, they look pretty much similar except for that of the micellar formulations, which is pretty obvious because it's not an emulsion, it's a micellar formulation. And also there is a significant difference in the particle size for that of the F4, which is actually a heterogeneous Q2 equivalent formulation from that of the F1. Particle size distribution of the emulsion globules is actually a very important critical quality attribute. So it is actually determined by calculating the span values. If the globule size distribution of the emulsion product is unimodal, then span value calculation will do. If it's a bimodal distribution, then we need to perform a population bioequivalence study. So if we can see here, the F4 Q2 equivalent formulation has a significant difference in globule size when compared to that of the F1 or the RLD. This is because the F4, the heterogeneous formulation that we formulated, is not subjected to microfluidization once it's formulated. So that's actually attributed to the significant difference in the particle size here. Before getting into the details of the IVRT technique, let's try to understand the basic differences between a quality control dissolution and a biorelevant dissolution method. So here I would like to compare our method to that of USP4. Of course, every method has its own pros and cons. USP4, the method parameters can be optimized easily. T10, T50, and T80 can be determined. On the other side, because of longer sampling time point, stability issues may arise, and it is very minimally biorelevant. Whereas our method, the method parameters can be optimized easily. The sampling time is as early as 1 to 2 minutes. It is very biorelevant in terms of flow rate, volume of the sample that we collect, perfusion medium, and the sampling time. Of course, T10, T50, T80 determination is very difficult, and we need to have a robust analytical method for doing this. So now, being said this, what we believe is a combination of a quality control dissolution and a biorelevant dissolution would actually accelerate and also improve the confidence in the generic product that is being approved. This is a cartoon that shows the working of our instrument. As you can see here, here is a syringe pump with a syringe fixed to it. The vial here has the emulsion in it, which is stirred and maintained at a given temperature, and there is an autosampler here. So the syringe is filled with the perfusion fluid or the release medium, which actually flows through these tubings, and it reaches the probe area where the actual diffusion occurs. And at a given time point, the samples are collected with the help of the autosampler here. So this is the depiction of a typical microdialysis instrumental setup that we have in our lab. Now, these are a bunch of factors that we actually consider to optimize, which are pH, membrane type, dilution ratio, membrane cutoff, flow rate, and perfusion fluid. So we actually ran the experiments in triplicates, and when we finally optimized the factor, we moved forward for performing the actual experiment. Now, if we take a look at the release profiles here, actually to simulate the differences in the manufacturing processes, and also to make sure that the probe or the membrane that we are using in the probe is not acting as a barrier for diffusion, we decided to formulate a variety of formulations here. So if we take a look at the release profiles, the RLD and the Q2 equivalent F1 formulation, the release profile is pretty similar. Micellar product and also the F4 Q2 equivalent formulation, which is a heterogeneous formulation, they are significantly different. Of course, the half strength and double strength emulsion formulations are also significantly different from that of the RLD or the F1 formulation. So the point that I want to make here is the approach that we used is not just able to validate the method that we develop, but also the instrument. So it's like we are able to leap 2 hurdles in a single jump. After understanding the release profiles, we conducted a statistical test, which is called the Wilcoxon test. This test is actually recommended by the US FDA to compare the equivalence for topical products. So when we compare the RLD with that of the Q2 equivalent F1 formulation that we developed in our lab, they actually pass the test. But when we compare RLD with that of the other products or the other formulations that we developed in the lab, they tend to fail the test. Moving forward, we actually tried to do an in vitro in vivo extrapolation. So to actually predict the drug absorption from the human cornea, what we tried is we tried to extrapolate the release obtained from the probe surface area to that of the human corneal surface area. So when we plot RLD with the in-house Q2 equivalent test formulation, which is F1, the plots tend to overlap with each other. In conclusion, the microdialysis method was developed and optimized, and the different parameters such as the composition of the dissolution medium, pH, molecular weight cutoff of the probe, nature of the membrane, dilution ratio, and the flow rate were optimized. The final optimized conditions were using a 10 millimeter length of polyether sulfone membrane probe with a molecular weight cutoff of 35 kilodaltons. The perfusion fluid has 1% albumin with 0.1x PBS at pH 7.4 with a flow rate of 4 microliter per minute and a sampling frequency of 2 minutes were optimized, and the experiment was conducted at 35 degrees centigrade. These conditions were found to be the optimal, and the sameness of the RLD to that of the in-house developed Q2 equivalent test formulations, which is F1, is proven by both statistical test and also the IVIVE experiment that we conducted. Now, an important point that I want to make here is the method that we developed is actually able to prove the sameness of same formulations and is also able to catch hold of pitfalls of the formulations that are different. Moving forward in the future directions, we would like to standardize the microdialysis instrumental setup that we have, and also we would like to develop a semi-automatic or an automatic instrumental setup so that this technique and this instrument can be applied across labs and in the industry. Now, I would like to take a moment to acknowledge US FDA for its initial funding, Department of Pharmaceutical Sciences at Texas A&M for their continued support, and DRPI for giving me this wonderful opportunity to present my work. Thank you one and all once again.